Biblical DNA here. New study. Human migration from the Levant in Arabia into Yemen since last glacial maximum. Now, it was funny when this actually dropped. I was on X with an individual that was debating of what I was referring to as far as when the experts are saying the proof of Semitic expansion of languages started happening. And I was telling him the experts were saying the early Bronze Age. He was saying Neolithic or Epapolithic with the New Teuton culture. I'm like, no, 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 no. It's early Bronze Age. And this paper goes into that. It just highlights exactly what I was talking about. I'm like, look at here, telling you the exact thing I'm talking about. And then he tried to say, oh, no, no, no. The Yemenites carry E1B1B. So it proves what he was saying. And I'm like, no, 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 no. This paper is saying that they're J predominantly. It was just hilarious. But anyways, let's see what this paper has for us. While a broad consensus about the first successful migration modern humans out of Africa seems established, the peopling of Arabia remains somewhat enigmatic, identifying the ancestral populations that contributed to the gene pool of the current populations inhabiting Arabia and the impact of their contributions remains a challenging task. We investigate the genetic makeup of the current Yemeni population using 46 whole genomes and 169 genotype arrays derived from Yemeni individuals from all geographic regions across Yemen and 351 genotype arrays derived from neighboring populations, providing regional context. Principal component analysis shows stratification between Yemen districts, but also with respect to nearby populations. Yemeni, other Arabian, and Bedouin samples form a continuum towards the populations of the Levant, whereas East Africa and India appear strongly differentiated. This finding is further supported by higher principal components, admixture, and haplogroup analysis, and F statistics. Moreover, two reference linkage disequilibrium decay estimates are most significant for Yemeni admixture from an ancient northern influx up to 5,220 before present from Palestine and East Africa 750 before present. We show that the initial gene flow into the Yemeni populations of today came from the rest of Arabia and the Levant, and a less substantial and more recent genetic impact into coastal Yemen from East Africa, particularly. Okay, now this is what we wanted to know of exactly where did the Yemen population come from? Where did those Y-DNA J1, why did it get predominantly in the Yemeni population? That's what we wanted to know, right? Let's go to it. Let's get that Bronze Age real quick on. All right. We have ancient context. Figure S8 shows Yemen samples in the context of ancient samples. The Sheba district that includes samples from Marib, Jawf, and Bada exhibits the greatest similarity to ancient Bronze Age Jordan, Israel, and Syria slash Abla. Read it again. Greatest similarity to ancient Bronze Age Jordan, Israel, and Syria, Ebla. And do y'all know what the most dominant haplogroup, Y-DNA, from these populations are? It's J1P58. Yep, I say it again. It's J1P58. The same haplogroup this individual on X was debating me about. And I'm like, this isn't that serious, my guy. I am dropping the Lazarus it L 2022 on you right now. I'm telling you, it's exactly what I'm saying. He's like, no, no, no. It's epipaleolithic Natufian culture. I'm like, no, bro. It's what these experts are saying. Just listen to the experts. If you got new data that contradicts, you publish your research at a university and you debate these scholars. If you can't do that, what were we talking about? I'm just saying. All right. Let me get that other part. Here we go. All right, y'all. I'm gonna read this whole paragraph that includes the Bronze Age because this is important because it goes into the Semitic languages, right? It says the limited Northern Arabian Levantine ancestry observed in the southern Yemeni region of Aden was likely diluted by the flow from East Africa. The findings from Beida clearly illustrate the absurd granted a northern Arabian Levantine meeting a southern East African genetic gradient. For decades, the prevailing literature has attributed cultural and linguistic similarities between the Levant and Yemen to multiple south-north population migration waves from Yemen to the coastal Levant, particularly in the aftermath of the bursting of the Marib Dam during the 2nd or 3rd century CE. But recent evidence by Al-Jayed and others document an Eastern Levantine genesis of classical Arabic supporting the narrative presented here in previous arguments proposing cultural and linguistic flow from the Levant to Yemen. From where, y'all? From the Levant to Yemen. These findings are also supported through the Bayesian phylogenetic analyses by Kitchen et al. Estimating an early Bronze Age origin for Semitic languages in the Levant. Now, if y'all been keeping up with family tree DNA and the Southern Arc, they're estimating that J1P58's expansion mutation started in Syria. That's what they're saying. That's what the experts are saying. That's what Lazarus et al. 2022 said in the supplementary material. And that's what family tree DNA is also saying. So anybody wants to debate the experts. They're telling you this. That's what they're saying, not me. That's what they are saying. Let's get into the haplogroups, right? J1. Let's see, here we go. We got this type of regional diversity can drive the genetic structure differentiating between these regions, offering insight into the broader history. Previous studies using Y chromosome analyses have shown that populations within the Arabian Peninsula, including Yemen, show some degree of genetic differentiation. Y chromosome haplogroups and YSTR markers analysis also demonstrate genetic diversity between Arabia and North Africa on the one hand and between Arabia and the Levant on the other. The Y haplogroup dispersal J1 and J1E in particular provides evidence of population movement between the Levant and Arabia dating back multiple millennia. However, mitochondrial DNA analysis presents a contrasting perspective showing less mtDNA haplogroup differentiation between Arabian populations and North Africans, but greater difference in mtDNA haplogroup distribution between Arabia and Levant. Finally, the combined autosomal DNA, Y chromosome, and mtDNA results provide extensive evidence for dynamic and multiple back and forth migration events between Arabia and East Africa dating back to at least 12,000 years before present. Let me get the other J1. So this is the phylogenetic tree for Y chromosomes derived from the 46 fully sequenced genomes is shown in figure 5. Haplogroup J1 is predominant throughout Yemen with high diversity. High diversity. High diversity. And yes, we do see mutations of the Natufian culture with the E1B, 1B, 1B, and also the Levant pre-pottery Neolithic B with E1B, 1B, 1A. So EM78 and EM123. But mainly, who do we have? Predominantly J1. It says the Y haplogroup distribution among the various Yemeni groups is strongly dominated by haplogroup J1 reaching almost 100% in certain regions. And the second most frequent Y haplogroup is E1B. These two haplogroups are the most widely spread Y haplogroups in Arabia and the rest of Southwest Asia and are highly suggestive of strong gene flow between these regions and Yemen as observed also in the PCA and admixture results. Not only do we observe a high diversity of J1, but it is also present predominantly in less admixed regions, indicating that it must have dominated the region for a long period. For how long? For a long period. These results add to the body of evidence suggesting that some of these haplogroups have been introduced to Yemen more than 5,000 years. How many years, y'all? More than 5,000 years. Hence, the level of observed diversity shown here. The predominantly African mtDNA-L haplogroup is also well represented in Yemen. 3 of the 5 sequence samples from Yahuda had a L2A1 mitochondrial haplogroup, the most widespread haplogroup in sub-Saharan Africans. About 1/3 of the Yemeni sequence samples belong to the African haplogroup L, suggesting a large African matrilineal influence that paints a different picture from what is observed from the mostly Southwest Asian Y haplogroup predominance in Yemen. These observations are not surprising given the historical trade networks that existed between Africa and Yemen. The African-specific mitochondrial DNA haplogroups most likely reached Yemen from East Africa and remained highly represented in Yemen through isolation. Migration between Yemen and East Africa has been very well documented since the 7th millennium BCE. In addition to trade of incense, spices, and other goods between the Old Kingdom of Aksum and Ethiopia, slave trade constituted a major part of moving people from East Africa to Yemen. I think I want to save this for the conclusion. I'll go back into this in the conclusion. I wanted to jump into the haplogroups, the distribution of the Y DNA and the mtDNA, right? And as you can clearly see, what I see, what this individual on X was debating about is the distribution of J and J2, or J1 and J2 amongst Yemen, the Yemen population, right? And you see all these Y forward slashes down here. These are all Yemen countries or cities. If you're not tracking these cities, they're all in Yemen, right? And you can clearly see which haplogroup is the most dominant in the Arabian Peninsula, without a doubt. In Yemen, which haplogroup is the most dominant? Y DNA in Yemen, it's J. I really don't know what he was arguing about. And I'm like, bro, I just clipped this for you and you still cannot understand it. Somebody else was trying to help me out, explain it to him, and he was still debating it. I'm like, this guy's just trolling. But it's so funny because he has people that I follow that are the experts following him. And I'm like, wow, if the people that I follow, that I, respect, are following this guy, they haven't seen what I'm capable of doing. Like, they have no idea who I am, because obviously I'm just going to spit out the truth and the most recent research, which is going to refute what this guy's saying. But anyways, one user from Yemen told me that why you see Yadu that so high in E1B1B is that this is a population that has a heavy African component, meaning a lot of its population came from Africa, East Africa, and that they aren't really like Yemenites, like they're not really Yemen, Yemeni. So they're not, I don't know how he explained it to me, but because he's obviously an Arabic speaker, but he was saying that these aren't really Yemen, like part of Yemen, but they're considered Yemen because they're living in Yemen. So anyways, you can take it for somebody that lives in Yemen. I'm not Yemeni, so hey, if you're Yemeni, please write in the comment your thoughts and concerns about this, or what your feedback. I'll much appreciate it, and I'm sure the people that watch this video will also respect and appreciate your feedback. All right, let's go to the mtDNA. So as we can see, in Ethiopia, Kenya, Niger, which haplogroup mtDNA dominates the most? It's without a doubt L, right? And so also in Yemen, we see it dominates there too. But like I said, this individual is saying is that these people aren't like Yemen, they're not like, these people are more recent. That's what he's arguing. That's what they're arguing. So again, if you're Yemeni, please, in the comment section, if you have more feedback to this, it'll be greatly appreciated. All right, let's go to the admixture graph, right? And I'm leaving this video separate too, because I know some people, they just wanna they need to understand according to a comment that I received from, I believe it's Atten, if I'm not mistaken. Let me see real quick. I believe it's Atten that made the comment that the community, a lot of people in the community cannot read admixture graphs, right? So I got y'all. That's exactly what I'm gonna do. I'm gonna show some admixture graph. I'm gonna explain this, and I'm gonna separate the video. Figure 2, the admixture graph from the Yemen study. The admixture graph from the study provides a nuanced look at the genetic diversity of populations within Yemen and their connections to broader regional ancestries. This graph visualizes the genetic diversity of individuals from populations within 3,000 kilometers of Yemen. Each colored segment in the bars represents the proportion of genetic ancestry attributed to a specific ancestral population. With K equals 5, we identify 5 major genetic components reflecting the region's history and migration patterns. The Hadramaut and Hadida districts show the greatest genetic mixture, highlighting their historical role as crossroads for trade and migration. So to further understand the admixture graphs, we have to understand the components of the graph. The populations, which is the clusters, is represented by colored bars or segments in the graph. The proportions is the length of each colored segment, and an individual bar reflects the proportion of genetic ancestry assigned to a particular cluster or population. K values. The K to the left represents the number of ancestral populations assumed in the analysis. Each individual is divided into proportions based on the inferred ancestry groups. Now, what does the K equals 2 through K equals 5? What does that mean? You may be asking yourself. K equals 2 is the population is split into 2 ancestral groups. This is often the first level of detectable genetic differentiation. For example, one group might represent populations with more sub-Saharan African ancestry, while another group might represent populations with Levantine or Arabian ancestry. The admixture graph shows 2 colors, and individuals might begin to show proportions of these 2 groups. K equals 3 adds a third ancestral population to the model. Typically captures more subtle differences, such as a third influence from East Africa, South Asia, or an isolated Arabian population. Populations like Hadramaut or Hadida with diverse histories may begin to show more complex admixture patterns. K equals 4. K equals 4 introduces a fourth ancestral population. This could represent further regional differentiation within Yemen or neighboring areas, such as distinct Arabian Peninsula populations and an Indian Ocean trade component. Coastal populations might show higher proportions of new components due to maritime trade influences. K equals 5. The model identifies 5 distinct ancestral populations with the lowest cross-validation error. That's critical. These 5 groups provide the most accurate balance between complexity and explanatory power for the data set. For this study, a clear distinction emerges between Southwest Asia, Sub-Saharan Africa, East Africa, and other influences. The study emphasizes K=5 because it captures the major genetic influences without unnecessary complexity. Population ordering. Populations are often ordered geographically or according to specific criteria, e.g., Yemen governates in your graph. Regional groups, similar ancestries, or genetic diversity within regions can be identified by common patterns or mixtures in the graph. Interpreting the example, governates grouped into districts. Aden includes Abian, Dali, Lahid, Azal, Amran, Damar, Sada, Sana, Shiba, Baida, Jawif, Marib, Hadramat, Shabwa, Hadada, Haja, Hadada, Mawit, Halajanad, Abe, Taiz. The key observations are noted with the strongest diversity in Hadramat and Hadada, suggesting these regions may have experienced extensive genetic mixing. Validation and error. The lowest validation error occurs at K=5, indicating the most accurate depiction of genetic diversity for this analysis. The color patterns. Each color corresponds to a genetic component. If an individual shows multiple colors, it means their genetic makeup derives from multiple ancestral populations. All right, y'all. I appreciate y'all watching. This been Biblical DNA. I'm out.